history in this area than other areas of behavioral mechanisms. Not surprisingly, I mean, what we're talking about is we're talking about the sort of the neuronal basis for behavioral expressions. And just like, behaviors always have genetic and environmental components, just like behaviors have developmental components. Well, behaviors are like any other trait. And the sort of the action of any trait is associated with neurological structures. And this we'll see is sort of a reality for multicellular organisms, obviously, but it also the same basic structure and the same basic mechanism occurs within unicellular organisms. So the same apparatus is there, even though they would never have something like a structural neuron. So what we're doing when we study neuroethology is we're studying the neuronal mechanism by which behaviors are expressed. And increasingly, how behaviors influence that neuronal mechanism. Can it make sense? Okay, so I'm going to add also a little history. A lot of these slides in today are some of the first illustrations I ever did. So you're going to see some really low quality, but I love them because they're still sort of my first adventures into animation and visualization. Cool? And maybe you could have this as a game, try to pick out which ones are which. Make sense? Yep. Okay. So, neuroethology is one of those sort of first level functions for the expression of behavior. And I kind of like this image right here in terms of the hierarchical arrangement associated with neuroethology. And you could really go in either direction. So a lot of times in neuroethology, we think about the behavior. And I'll put on this video of billfish and lateral lines. And one of the things that we might see within billfish is the expression of intraspecific aggressive behavior, where one animal is being aggressive to the other animal within the same species. We typically see this in things like male-male aggression, to which we have a variety of different behaviors that are associated with that. Right now, what we're seeing is something called a lateral display. It's when two fish will swim next to each other, side by side. And there's a variety of explanations for that in terms of the adaptation. They can evaluate whether this is a contested fight or not. If the animals are fundamentally disproportionate in size, they tend to just depart, and sort of the smaller animal just leaves the scenario. If they're relatively equivalent in size, then they may escalate the behavior until eventually you might see something like a fight, where a fight is very rare in terms of intraspecific competition. Within these lateral displays that we're seeing, there's a whole variety of things that are going on. There's the alignment of the body. There is the structure of the tail beating. We actually just saw that. There's the erection of the fins, and I'll go back a half a bit so we could sort of see that come up, where we have sort of the entire display of the sail fin. We have the spread of gill covers, and we have the intensification of colors as it can actually change a little bit of what their surface color looks like. So this aggressive display can be broken up into different parts of it. And then the different parts of the display, obviously, as we talked about, amalgamated behaviors have different components. And we'll restart with all that fun stuff. And then when we actually look at the erection display, we have additional pieces that go into it. The actual movement of each individual fin. Let's see if we can get that nice. Oh yeah, we're about to hit it. The erection of each individual fin, the contraction of the muscles that allow those fin rays to actually move away from the body. And then the base root of neuroethology would be the neuronal controls to those motor units. So we can look at this in terms of a bottom-up approach, where we're starting from the neuron and looking at how the neuron influences the different levels and scales of morphology to the behavior, to the overall amalgamated display that the animal is trying to do. And we could also go from the opposite journey, going from the aggressive behavior all the way down to the neuronal mechanism. Cool? So there's going to be some very, very bio 1 type of reminders in nerves, because these structures are important to understand how ridiculously simple system can do something as complicated as we see within like the billfish. And neurons are awesomely simple. So what are we talking about? Well, we're talking about, dendrites, extensions away from the overall cell body. We're talking about the cell body, and then sometimes we have an elongated axon. Within that, we can have different Schwann cells and mammalian sheaths that can help with the transmission of a signal until we actually get to the connection between two different neurons, as in an intermotor neuron or an actual source or motor neuron. And therefore coming to a structure of a topic. When we have an action potential, what we have is a differential charge of the neurons starting at one of the synapses, leading down from the dendrites through into the cell body and then going out the axon until that information is communicated the other side. There are different classes of neurons. And most simply, we could have sensory neurons where we have the dendrite and axons respond to a non-neuronal stimulus. So they're responding to information from the environment. And remember that information, when we say the environment, means both the external, which is what we tend to think about, but it can also mean the internal environment, what's actually going on inside the body. But it's responding to something in the environment that triggers the nervous system. We have whole classes of interneurons, whether those interneurons are actually partially responding to in the brain or other parts, maybe ganglion, or even just sort of dense nerve areas, where the dendrites are connected to other dendrites, and therefore the information is shared from one cell to another. And then we have motor neurons, where the dendrites terminate at an endpoint, such as a muscle. And therefore the information of the neuron is translated into a response of the effector. Gonna make sense? Good reminder? Yeah. So neuronal signals, they have two ways in which they can be communicated among cells. One is through electrical synapses, where we'll have things like a gap junction. We have a single action potential that go right across from one axon directly through to the dendrite of a second cell. And then we have another structure where we have a chemical synapse, where an action potential goes along the axon, terminating at the dendrite, and then is translated, not from an electrical signal, but into a chemical signal, which is then picked up on by receptors until the next action potential or until the response is made. So far, so good. So all we're doing right now is just laying down some foundation before we can actually start to translate how we go from these so freakishly depauperate and simple mechanisms to the complexity that we actually see neurons able to accomplish. So a reminder of an action potential, and just an FYI, man, people bomb this. So that is my hint to you. You might think you remember it, but maybe based on evidence of previous students, not everyone does. What we're looking at here is we're looking at an overall differential in terms of the membrane potential. And the membrane potential is where this electrical signal goes across the surface of a cell membrane. And what we have in an action potential is a sudden shift in that membrane potential that changes the charge of the inside of the cell relative to the outside of the cell. And in changing that charge carries information. You can almost think of it as well, I don't know. What are we all on right now? All of us, in one form or another. What are we on? What are you looking through? What are we looking through?
Like a laptop, the camera?
We're looking through a computer. And the action potential runs really the exact same way that a computer runs. And a computer runs based on a binary system, zeros and ones, ons and offs. Action potential is the fundamentally same process. You can think of an action potential as a 1. And every time there's an action potential, you got a 1. And when there's not an action potential, you have a 0. And therefore, much like a computer operates in terms of the sequence of 1s and 0s, This is how an action potential can operate in terms of nerves. Now, what we're looking at is we're looking at this change in membrane potential, where membrane can shift from a positive to a negative environment, negative to a positive environment. And we're going to look at this in two ways. We're going to look at this graphically, where we're going to separate out this action potential into 5 different parts. And then we're going to look at this across what is actually going along the typical cell. And this is highly, highly generalized. This is probably the image that you were presented in terms of bio 1, but there's a lot of variation from one nerve, one system to another nerve, another system. But this is a good general description. And what we have is we have sort of a stimulation phase where we are generally negative in terms of the membrane potential. The inside of the membrane is negative relative to the outside of the membrane. What happens during the stimulus phase is that there is an opening in sodium potassium pumps where sodium crosses the membrane. Sodium being positively charged crosses into the membrane. We start to see a slightly more positive membrane. In this generalized case, the membrane potential is still highly negative, but we have some positive ions going across. We then get a rising phase. Basically, we hit a threshold, and the threshold is cell specific, where at this threshold, all of a sudden we get an onrush of ions across the membrane, where in front, we have a negative interior. Once we hit this threshold, we have all of these sodium ions rushing inside the cell. And what they do is they can create a positive environment inside the cell. And this charge shifts very quickly from a negative charge to a positive charge. In computer language, a 1. What we get is we get a refractory phase. Once this gets to a very positive environment, there is a flip. And what happens is potassium is now rushing out. So we start in a negative environment with small positive ions coming in. We hit a threshold. Now positive ions are rushing into the cell. Once we are supercharged, we get an exit of potassium ions. And basically, we now have positive ions rushing out of the cell. If we look at this sort of graphically, we have an increase of the membrane potential that all of a sudden becomes very positive. And then as quickly as it becomes positive, it now becomes negative. And then we hit an equilibrium where basically things kind of settle out. And we're back to that largely negative interior cell. And there's sort of a pause because there is an overshoot, typically, in terms of there are more positive ions that leave the cell than would be appropriate for the equilibrium. The next action potential cannot start in the typical situation until this equilibrium is met. So we have this overshoot that sort of delays, slows down the response until we can finally have a second action potential, where the process is exactly the same. Positive ions drift in, the membrane becomes a little more positive. Some threshold is hit. And the threshold just describes when we get the flood of ions rushing into the cell. That overshoots the membrane potential. Again, cell by cell, a little different, to the point where we get a refractory response, and we now have positive ions rushing out. These positive ions rushing out, this also overshoots. Now we're too negative, and it has to get back into a little bit more positive before we can hit an equilibrium. That's the process. Now let's think a little bit about the logic. Okay, so, oh, heck. I gotta find my participant list. I didn't randomize the class today, so, we're gonna be doing Taylor. You're up first. Okay, so in his first stage, the stimulus stage, we open the sodium potassium pump and sodium starts to cross in, but it's not an active process. It's a passive process. Why does sodium enter into this negative environment inside the cell?
Because of the differences of charges.
Perfect. Perfect, perfect, perfect. We then hit a rush and we get a ton of positive ions running into the interior of the cell. So, Olivia, why do these potassium ions leave and rush out? As fast as we get this spike in the membrane potential, we see refraction in the membrane potential. Why do we see this rushing out of potassium ions?
I guess because the other ions entered.
And what happens when you get positive ions next to each other? Like lots and lots of positive ions.
They're gonna repel each other.
They're going to repel each other. And what's the only out? What's the only exit?
To leave the membrane.
And through what?
Sorry, it broke up whenever you said that.
Through what?
Oh, backwards to the pump then?
That's the only place to leave. They can't go sideways because they're still sort of messing with each other. So really the easiest place for these ions to move, and it's a passive process. It doesn't need any sort of energy and it doesn't need any sort of intellect. Really the only opening is to go back through the pumps. So the potassium ions rush out through the pumps. Now we're too negative. What's going on here? When we're too negative, Megan.
It like spreads out it moves the positive ions to like counteract the negative ions, I guess, or
Yeah, and that's a fair statement. It's not very visual in here. And I didn't draw this with enough sort of negative ions to sort of show that. So you described it well. There are too many negative ions, so they basically just have to diffuse out of it, which is why there's sort of a proverbial or essentially a slower response to get back up into equilibrium. But once we get back into equilibrium, you can repeat the process where sodium potassium pumps open up. Sodium moves in because it's attracted to the negative environment inside the cell. A threshold is hit. Now an explosion comes in. Too many ions that are positively charged are next to each other, so they're repelling each other, which allows those potassium ions to rush out of the sodium potassium pump until it overshoots and settles back through diffusion into an equilibrium. Now, if we're going to treat this as analogous to a computer, what is this? In the binary language of a computer. Millie.
Can you repeat the question?
Sure. In this part of the graph, where we have the rising phase and refraction, really right up here. In the language of computers, what is this? In the binary language of computers.
Would it be the peak?
It would be the peak. In language of computers is ones and zeros. So we might think of this as a one, i.e. on. And Ella, what is this period?
0.
0. So an action potential really kind of reads kind of like a computer language. On, off, 1, 0. Does that make sense?
I have a quick question.
Yeah, far away, Connor.
So in regards to this, if we're talking ones and zeros, the between area with a 2 and 3, is that technically still a 0? Like,
I am making a gross metaphor, not in a sense gross like is ew. I'm making a gross metaphor in the sense of like it's kind of a hyper generalization, but
yeah,
it helps to
sense.
think through, and this is why I sort of make this metaphor. 2 parts. One, neurons are as simple as I'm stating. They are on and off. Depending on the structure of the synapse, they're literally just a continuation of on. They're a really, really simple system. Much like a computer is a really, really simple system. A computer is binary, 1 and zeros. But based on how we organize that binary information and how we organize this process of act potential on, act potential off, we can start to create the ridiculous complexities that you can appreciate in the computer in front of you. And you don't have to know anything about computer to appreciate the fact that we are all literally in different environments. We are all literally changing the way in which a signal is being provided in terms of a visual signal like you are in your own space, that's getting translated into a binary language through your lens, that's getting transmitted over wavelengths if you're using Wi-Fi, or if you're using a direct connection cable through, electrical circuitry, that then gets retranslated in your home computer, that then gets organized in a way so these ones and zeros are now communicating in a way that we see a visual representation of that image. I mean, honestly, if you're not going WTF, I don't know who you are. And I don't mean, I'm sorry for anyone who's confused, I do not mean Wisconsin Trade Federation, which for a long time had the URL www.wtf.gov. That is true. That's no longer the case. So so it's a gross metaphor. And if the metaphor confuses, I apologize, but that's the point behind a metaphor, to sort of help us recognize that in many ways, the same way we don't really fully understand how computers can operate using the binary language to do the things that it does, neurons are kind of the same way. And it takes time to really sort of pull apart maybe small little pieces and get a sense of that, to then sort of increase out the complexity for what is capable. And some of the mechanisms that we'll talk about today are intentionally really simple because, one, we can measure them because they're simple, and then two, because man, this stuff can get confusing quickly. I mean, like so awesomely confusing. Great question, Connor. Others? All right. So, this shouldn't come as a surprise, but being able to respond to your environment is key. And even for something like a protist, which is a single cell, it's got to be able to respond to its environment. And really, it shouldn't be a surprise that the environment inside a cell is maintained differently than the environment outside the cell. And as a result, something like a protist can take advantage of that to essentially make its entire body one big nerve. And while when we talk about the generalized nerve and something like a human, we're talking about sodium potassium pumps, what you have in terms of a protist is you have a potassium calcium pump. And what happens here is that you have a differential cell membrane. We have a different charge on the outside than you have on the inside. The cell is more negative than the entire environment. And what happens when the protist, in this case a paramecium, is hit in its anterior end, this potassium pump opens and potassium rushes out. And that causes the protist to move backwards. When the protist is hit in the posterior end, you get a depolarization of the membrane. Calcium rushes in. So the interior, this area of the membrane, is more positively charged, and that gets the paramecium to move forward. And if you think about existence from the perspective of a paramecium, that's really useful and fruitful information, where a lot of the things that you come into contact, which are bigger than you, they're dangerous. So if you come in contact in your front, move back. And they can do that because of the membrane being stimulated and potassium rushing out, and this area becoming more negative. If you're contacted in the posterior end, move forward. And they do that by calcium rushing in, this area inside the cell becoming more positive, and into a signal to move in that direction. And as we remember from, bio 1, bio 2, we're talking about cilia and different ways they move. Now, two things about this video. One, this video is coming from the perspective of amoebas. Whoever took this video loves amoebas, but they're fantastic paramecium all over the place. As you remember with amoebas, they engage in phagocytosis. So they basically just sort of cover the body of a food item and then they consume it. What I want you to do is not geek out on the amoeba, which I know is going to be hard to do. but focus on the parameciums. And if you also geek off on the music, well then good for you. It doesn't do for me, but and watch this one for a bit. You can see the cost of failing to redirect in that all three of these amoebas, well, all four of these paramecium are presumably going to get hit. Neat. So if the animal is able to move away from contact, either by going backwards when the contact is in the anterior end, or by going forwards when the contact is in the posterior end, that increases the likelihood that the paramecium doesn't find itself in this situation. Make sense? Now this is getting into smooth jazz, which is the only reason we're not going to see the rest of the video. So one of the early behavioral genetics studies was a knockout study, where what the researchers did is they basically developed a group of paramecium where a gene called PON was knocked out. The typical response to the paramecium is when it hits an object, the anterior end, calcium is depolarized and the animal moves back. Oh, by the way, just so you know, that's your paramecium. This right here, the line, that's a predator. Where going through that line is going through the predator. I told you, these are like first generation animations for me. And by contrast, this is PON. And when PON is knocked out, what happens is the calcium is not permeable, there's no depolarization, and these animals are consumed at much higher rates. Questions? Even though, of course, use of the word animal is probably inappropriate. These protists are consumed at much higher rates. So neurology is obviously, and we think about it much more clearly in terms of multicellular organisms. There's been a lot of work recently on electric eels in the Amazon. Two things in this. Please pay attention to the video of the animal, but I also want you to pay attention to the clicks of the animal. What the animal does is it sends out electrical pulses through its neurology that create an electrical field around the animal and that allow it to see its environment very similarly to the way bats use echolocation to see their environment. Each of the clicks is basically an electrical pulse. So Corlin, can you describe a little bit of what you're seeing and or hearing?
So in that specific thing a couple seconds ago, you kind of saw the eel swim a little bit past the fish and then quickly snap to its right and capture the fish.
Okay, I think it's a really good description. So, whoops, we'll go back. I'll take a look at that real quickly. So I think it's a really good description. So it kind of swims a little bit past the fish and then sort of quickly turns towards the right and captures the fish. I don't think I see Julie. Shyler, what are you hearing in this one?
I don't know, it's just me, but like I only heard clicking for like a little bit and then the video went silent. I only heard in like the beginning, but I just heard like they were kind of like clicking noises.
It's clicking noises. And why don't I replay the clicking noises? So first I'll apologize for that. When these videos are made, they're not made with all of the sensory input. So this is a video where they took the video and then what they did is they matched it up with audio recordings that they have elsewhere and they aligned them to match what should be going on. And that's why it sort of disappears. You know, the opening of the video, they want you to hear that clicking noise. The part that we're looking at here, they didn't layer it in terms of audio, but the animal is clicking. And then you get a little clicking right there. So what do you hear in terms of that little piece right before the prey was captured?
I mean, yeah, like I kind of just hear click, like maybe it's like something that they do so that the prey doesn't feel like threatened. I don't know.
Well, that's
So then he could just like
idea.
turn.
Let's not hit function first.
Okay.
So I'm going to come to you, and I always understand the desire to go into function. Let's first process what we have, and then we can sort of think through the function. So Sam, were you able to hear that clicking sound?
It kind of sounded like it got more rapid when I got closer, kind of like how it like the waves would bounce off quicker because it's closer to the object that they're looking for.
Well, let's hold off again on the function.
Okay, yeah, so like it sounded more rapid
It
because
sounded more rapid.
it sounded
Does anyone remember anything about sort of bats in terms of the way they echolocate?
I think it was about the same. Like when the bats echolocated, like the waves were shorter when something was closer to them, and they were a lot longer, and would bounce off certain objects.
Okay, so let's hear that. So we have this. And at this point, whoops, dear Lord. At this point, those clicks, those electrical pulses sound pretty even. At this point, as you said, it sounds like it's actually speeding up, and it does. So it's very similar to that sort of search phase versus approach phase versus the buzz phase in terms of echolocation. When in a bat, there's a regular call, you know, click, 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 click call. But as it gets closer to a prey, it increases the overall frequency of those calls until it actually makes contact. We get the same thing here. Cool. Does anyone know anything about these electric eels? Like,
Aren't their eyesight very poor?
eyesight's terrible.
Yeah.
So visually, they're not really going to find the animal. And maybe, and now we're just speculating, but Corley noticed at the beginning, it kind of looks like the eel goes past this, what I'm presuming is an archer fish, and then moves back and actually makes contact. And maybe that's the case. So the vision is very poor. Anything else we know about these guys? Well, one, the electrical charge is huge, very dangerous. And when they're at that last stage, they put out a very strong charge that fully stuns the prey. And if you are hit by that charge, it can knock you out. I mean, it is a very strong electrical signal. They live in the Amazon. Can anyone describe what the water of the Amazon's like?
Murky.
Murky. It's opaque, man. You can't see through it. You could put your hand in Amazonian water, and when you get to about your elbow, you're not really able to see your fingertips. So you have an environment that is very, very opaque. Eyesight's not going to do you much good, which is why there's so many electric fishes, presumably in the Amazon. There's so many species of electrical fishes. But these guys, electrical eels, they are their own breed. They're huge. Their electric charge is enormous. It is thought that one of the reasons that they have such a strong electrical signal is because they need to fully stun their prey. They cannot eat their prey if it's moving. And one of the peculiarities of electric eels is that they have lungs, but the lungs are in their mouth, and they're in the top palate of their mouth. So if you open up their mouth, you'll see sort of this membranous tissue, much like, think of our lungs. If you took out the outside layer, so all you were seeing were the alveoles and the vacuole passages, so it's just sort of ridges and molds. And are lung sensitive tissues. Well, you can ask yourself this: do we protect our lungs as humans? How do we protect our lungs from the outside?
Ribs.
We got these very nice sternum, good ribs. I mean, we don't do a lot of muscle attachment to them, but that sternum, those ribs provide a lot of protection for their lungs. These guys have their lungs in their mouth. Can you afford to have an animal moving around as you're trying to swallow it? No. Some of the thought is one of the reasons for the strong electrical signal is so that the animal is completely incapacitated when it's consumed. Amazing animal. Questions? Oh, and there was a recent study that came out maybe, I don't know, 2-3 months ago that showed that electrical eels will hunt in packs under certain conditions, which is just an awesome horror movie in the making. So maybe it will be 2030, 2032. We'll have a horror movie of animals that have electrical charges or villains that have electrical charges that walk around and hunt their human prey. I don't know yet. All right, so we talked about the synapses. And we have two main ways of doing it. We have electrical synapses, where the electrical signal, the action potential, just moves directly across what are called gap junctions, just little holes within the cell membranes that allow for that membrane differential to just continue from one cell to another. We also have a large number of chemical synapses where the action potential terminates at the dendrite, and that can release a class of synapses, which then make contact with receptors on the other side of the synapse. And then once a particular threshold is met in those receptors, the action potential can travel along the second cell. So we have a case of the electrical synapse where the action potential just goes directly from one cell to another. And then we have a case where the action potential, this electrical charge, just one, can terminate in a neurotransmitter. And then depending on the neurotransmitter, depending on the situation, may interact with any of a class of protein receptors. And then depending on those protein receptors, will lead to another action potential. Can it make sense? So I've given you a lot of stuff. So I want you to challenge with sort of the big question. How can we create so much behavioral variation? Learning, memory, plasticity, with such a freakishly simple mechanism. Neuron, action potential, on and off. Electrical synapse. Just maintain that action potential, lengthen it. Chemical synapses. And one of the things I didn't tell you is there are very few neuroreceptors. Give me some neuroreceptors, some that you guys know. Oh, we could play this game. I'm going to call you out and we're going to play it. Identify a neuroreceptor. And the first one who can't identify a new unstated neuroreceptor is the one who loses. So since you're on a computer, you can also start looking up information as I look over to my screen to see who the first person is going to be in this diminishing return game. And that is going to be Natalie. So give me a neuroreceptor.
I can't think of one from.
Natalie, I gave you so much time to get on that computer and start plugging in some stuff.
Okay. I think there was one from physiology that I remember that's called acetylcholine, I think.
Okay,
Yeah.
great, love it. Liz, give me another.
Dopamine.
Dopamine. That was kind of the first one I thought we'd go for because we're all so joyous. Twice a week when we get to come together in animal behavior and you are just flooded with dopamine. Yeah, so love it. Thanks, Liz. Carla, give me another.
Serotonin.
Ooh, love it. Another feeling that you must be just overwhelmed with right now because again, we're in animal behavior. That's a perfect choice. Thanks.
Norepinephrine.
Ooh, love it. Love it, love it, love it. Dakota.
Histamine.
Ooh, that's not what I love. I don't like that one at all, but I'm going to accept it. Excellent. Cindy, can you give me another?
Glutamate.
Great. And we're starting to tap out. There's, you know, depending on the organism, depending on the situation, 10, 20s or so. They're there. There's variation, but they're not hyper diverse. So we have a system which is the pop rate, action potential or nothing. Chemical synapse, all you're doing is just extending the action potential. Sorry, electrical synapse, all you're doing is extending the action potential. Chemical synapse, you have a limited number of neurotransmitters. And with just this limited architecture, we have the entire diversity of behavioral variation. Diversity of things that can be learned, remembered, the plasticity that organisms can show in terms of changing environments, both internal and external. How? So, great. So, Alex White, why don't we start off with you, buddy? What's one way in which an action potential can be used to create variation?
Well, I guess one of the things I mentioned in my group was like snakes. In order to constrict their prey, you would think they would need to extend potential throughout their muscles to last longer, and so maybe they're more accustomed to having maybe a more positive charge or negative charge, or keep that plateau a little longer than usual.
Oh, that's interesting. You know, and I like the idea. We don't really have examples where the length of the peak widens or sort of gets higher. Now, cell by cell is different, and you can have different types of cells having different thresholds and different responses, but it's kind of an up and down, but you're onto something that can create variation. So, and I like the use of the example within a snake as sort of a model for thinking through the action potential. Natalie?
So maybe like the peak goes up and then it goes down, but then it comes back up rapidly. Like we have a short period where it goes down.
It's a great
So you
option.
get that continuous like constriction, like he's saying.
So you can have a very rapid action potential, fast, 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 fast. Or alternatively, and what would be the alternative?
A great amount of distance, so slow and so go up and then go up.
Absolutely. And if you think about the bat echolocation and the electric eel that we just saw, it's still the same basic logic. You can change the latency between consecutive action potentials, making them very rapid order, and maybe that provides one type of information, or making it slow between action potentials. One action potential, then another action potential, then another one. Great observation. Casey, what about electrical synapses? How can that create variation?
I was kicked out for whatever just happened, so I feel like I missed it.
Well, it was the question I sent you guys out for the breakout room.
Yeah, I wasn't there for that. I got kicked out because my screen went black, so I had to restart my whole computer.
I'll bring you back in a little bit. Caitlin.
Sorry.
In my group, we were talking about how like certain stimulants can kind of affect like different action potentials, which are then affecting like how you feel. I don't know.
No, actually, I very much agree. What we're trying to figure out is how can an action potential, which is on and off, an electrical synapse, which just extends an action potential, and chemical synapses with a small number of neurotransmitters can do exactly what you just said.
I feel like they all kind of like talk to each other.
Let's think. Should this create a lot of variation or little when you have a system that's just on or off?
It seems like it shouldn't create a lot.
Shouldn't create a lot of variation. Okay, so let me ask another question. And I'm going to go to Heather for this one. Heather, our alphanumeric system, how many symbols do we have?
I think it's like 26 letters, and then you also have the numbers.
And how many numbers do we have?
I think 9.
I'm going to include 0, so 10. But perfect answer, by the way. That was rock star. Okay, so we have 36 alphanumeric symbols, right? Okay, is that a lot of variation or not? How many symbols does a more of a pictographic language like Chinese have to communicate?
Thousands.
Thousands. In fact, a moderately literate individual is thought to recognize some 20,000 characters. How many alphanumeric symbols do we have?
36.
Heather, you did so well before. Let me call back on you.
36 is what you said.
36. I'm going to count 0. Yeah, I like my zeros. I'm a little Mayan like that. So we got 36. Is that a lot of variation or not? It's not. It's not. And yet, is our use of language depauperate? Is it sort of limited? Is the poetry we say, I don't know, boring? Are the lyrics we sing unenthused? Are the conversations we have for dolgers? Why? How can we create so much variation with such a limited alphanumeric system?
There's so many combinations you can make.
There's so many combinations. And that's the same basic logic behind DNA. DNA, which has 4 base pairs and 20 codons. Because you can arrange them in different combinations, you really have infinite amount of variation. Infinite amount of variation. So nerves function differently. They're not like alphanumeric letters, but they have the same issue. And that is, there's not a lot of variation within the actual mechanism, but how they're utilized, the ways in which they're utilized can create tremendous amount of variation. So I'm going to send you guys on a break, but part of your break, I apologize, is going to be thinking about, well, how do we create all that behavioral variation with such a limited amount of mechanisms? One mechanism has already been identified and was really clever. You can change the frequency of action potentials. We can get one piece of information by having very rapid sequence of action potential, a different type of information by have widely spaced action potentials. And you can kind of see immediately, well, it doesn't have to be binary like that. You can have like a little segment of fast, slow, medium, fast, 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 slow, medium. And that combination, that sort of that nuanced arrangement of fast, slow, and medium action potentials together can create quite a bit of variation. Make sense? So we're going to think about electrical synapses, and we're going to think about those chemical synapses. I will see you guys back here. Oh, it feels like a good day. Come on back here at 11:55. Enjoy 14 minutes. We talked about action potential. You can vary the latency between action potentials. It's a great observation. How can chemical synapses maybe help us promote variation? I mean, all we're doing is just extending that electrical signal. So, Connor.
So I was thinking for like we're talking about chemical synapses, right? So on, I think slide 11, you had the difference like between like chemical and electrical with the neurotransmitters like emitting synapses, and different colors below, I believe, represent different forms of synapses. If you could have like different concentrations of the kinds of synapses like down there, like different like instead of it being like 2 yellows, you have like 5 yellows or like 27 yellows.
Okay, so that's
representing a different kind.
Just to be clear, these are receptors, protein receptors.
Okay.
So they're what the neurotransmitter binds to. But one of the things you could do, and it's a very good observation, is you can vary the density of different types of receptors. And one of the things I didn't state, so it's totally unfair, is the neurotransmitters are relatively limited amount of variation, but not the receptors. The receptors are highly variable and much more variable than the number of neurotransmitters. And they interact with neurotransmitters in different ways. So you can have like different combinations of neurotransmitters acting on a single receptor. And then because there's so many different types of receptors, you can have different types of information being communicated based on which of the receptors are responding. And if you remember when we talked about the monogamous rodent, that's a lot of what we have going on here. It wasn't the actual transmitter that was important in that system. It was where those receptors were located. And that's another piece. You know, where you have chemical versus electrical synapses located and what dendrites are associated with an electrical versus a chemical synapse can create tremendous amount of variation. So maybe the last one that I'm going to ask, and JC, I'm coming to you, buddy, and I don't see Nate, so I'm looking at you, JC, is how can an electrical synapse, this right here, how can that create some variation?
I would say like the amount that it looks like they like turn on and off, so like they open and close, I guess. And like the amount that they open and close.
Okay, so that's actually pretty clever. So they're an open closed system. These gap junctions don't have to be open. So when closed, the electrical synapse is going to stop at the dendrite. I wasn't even thinking of that, JC, but that was clever. Good. Another thing they could do is because they can extend an electrical signal, and electrical signal is a very fast process, they have the ability to basically communicate that information more rapidly to different endpoints without any complexity of the signal. That fast response. versus slower responses that you might use like chemical synapse for. So the neurons, not highly variable, but what can be done with them and how they can be done can be really quite variable. And the behavioral variation that we see from such a system, really, and again, I'm going to go back to that sort of overused metaphor. Strikes me as wonderful parallels between sort of the ways computers operate. Very simple system, but that simple system used in combinations can create the massive diversity that we use in terms of computing in the same way that this simple system used in different ways can create the complexity that we see in behavioral variation in the exact same way that a fundamentally depopulated alphanumeric system with only 36 characters or DNA with 4 base pairs, because you could use it in different combinations, can create all this wonderful variation. Does that all kind of make sense? And again, for me, maybe not you, but for me, it is still a, I mean, neurology pissed me off as an undergrad, made me so mad. It was too simple. Didn't make any sense. But it was kind of like genetics, which made me so mad. It's too simple. Didn't make any sense. But what has happened in all these cases is it's really how things are being utilized, when, at what rate, and what combinations that has become sort of the emerging story behind all of these. And that sort of variation that can be created, which is awesome. Questions? All right. So, squids. Squids are awesome. And what I want you guys to do is I want you guys to look at these squids, which are sort of in a mating group. And this video has been cut to suggest that there are stingrays coming by to eat the squid. And there is one squid that goes down in this video. So if you're a squid fan, I apologize. But squids have this incredible ability to retreat from a location. And they can do this by forcing air through a mental cavity. And when they do that, that pushes, I'm sorry, not air, water, forcing water through a mental cavity. And when they do that, that forces water out a very small orifice in a siphon, which creates almost a jet-like propulsion in the opposite direction. Poor squid. But you can see how in combination with their streamlined body, that collapsing of the mantle cavity to force out that water can allow that animal to move at exceptionally rapid speed away from any potential danger. I think the last few images were maybe the best in this regard. And that from a functional extent, obviously failure to do that leads to this small image. Now squids have been really maybe our first model organism, not only in neuroethology, but just straight up neurology. And one of the reasons for it is their nervous system includes a giant axon, which you gotta say with that voice. I mean, anytime you have the word giant in front of it, it's gotta be guttural. And what makes this a giant axon? Well, it's a cell. It's about 1 millimeter in diameter, which means it's a cell where you can literally see this axon, which is huge lengthwise, but also ridiculously thick with your very, very simple naked eye. Even if you have bad vision, you can still see this sucker. It's enormous. And you can see the axon diameter relative to other tissue within the membrane of the cell. And they use this in combination with other things in order to create a coordinated action in their mental cavity so that they can force water out from the posterior of the animal through to the anterior of the animal and out the siphon. In addition to the giant axon, we have basically, it can stimulate from it's a ganglion. So it's like a little secondary brain, a stellate ganglion, which is located right above the siphon. So really at the base of this mental cavity, and is associated with the control of the cavity's constriction. Make sense? Questions? Okay. So what I want you guys to do is I want you guys to hypothesize using what we know presently about how neurons operate in combination with a little bit of information we actually didn't talk about, and that is how the diameter of an axon can help regulate the speed of an action potential, to come up with a model, a hypothesis for the neurological architecture of a squid. We have the eyes up here, central brain is right through here. The stellate ganglion is a little bit behind, right near the base of the mental cavity. There's water held in this chamber. And for the squid to be successful, it needs to start pushing the water from the posterior end to the anterior end so that the water is forced out in a coordinated fashion through this siphon. Information, processing, additional processing, organization of the mental cavity, creating a wave of water that comes from the posterior to anterior, and therefore allowing that squid to move back. Using what you know about action potentials, using what you know about chemical and electrical synapses, using what you understand about terms of nerves and wide nerves versus thin nerves, where wide nerves can create a faster action potential. And you can kind of see where that is occurring because those sodium ions that initially go in, they're not interfering with each other because they're really far apart. So while the membrane is changing its potential, inside the cell, you're not getting conflicting ions that are trying to repulse each other. But when the nerve is really thin and those sodium ions move in, you got two positive ions that are right next to each other. And even though there still can be an action potential that occurs, there's some little resistance to the diffusion across the membrane. Make sense? So I'm going to open up the chat. And I'm going to upload our squid. The squid is now in the chat. You're going to want to download that. And what I would encourage you to do is have someone in your group share their screen. And then if you go to the options in Zoom, one of them is annotate. And if you open up annotate, you have the ability to draw, the ability to add text. And what you're going to want to do is you're going to want to draw out a hypothesized model for how to get information processed initially in the eyes, done something in the brain, done something in the stellate ganglion, carrying out through a giant axon and other axons to the mantle cavity so that you can get that collapsing from the posterior to the anterior end. And you're going to want to draw it out. And you can use text to then describe what's going on in each stage. You're going to want to label things. What type of synapse are you using? Are these thick axons, thin axons? What's going on in the stellate ganglion? You might want to provide a little hypothesis as to why. This all making sense? And then when you have this drawing that you're excited about, you're going to need to take a screenshot, because you're going to need to share it with everyone in your group. Because then we're going to come back and we're going to look at each other's models, and you're going to describe how you would hypothesize the neurological architecture to be designed for this system. And I promise you, there will be similarities, but there will also be differences as we all accomplish the same goal for this squid. And then there's the way the squid does it. But right now, I don't care about that. I care about how you think it would operate based on your understanding of the neurons. We good? We know what's going on? Take the image, share your screen, one person draw it out, make sure you put in information that describes it. And you really want that, because otherwise you got to memorize stuff, and I hate memorizing. And then when we get back together, we're going to share. And you might be the person called out. And you're in a group, but you still have to have the image and you have to share it out. Cool? All right, I'm excited. So, I can't find my mouse. There it is. There it is. I'm going to stop sharing, and I'm going to send you guys out to breakout rooms, and let's recreate. Okay. 10 minutes, 15 minutes. What do you need?
I feel like 10's good.
10. If you have any questions, give me a yell. I'd be happy to come into your room. See you in a bit. Set a timer for 10 minutes.
Okay, 10 minutes and counting.
Well, let's see is I went to screenshot the picture and all the words disappeared from our edit.
Oh no.
Well, you can still talk through it.
Yeah, we can talk through it.
I'll share the image and try to like fill it in while someone talks over it. All right.
Wow, it's so funny. We have to memorize what we said after Gunnel's saying, like, that's not gonna.
Okay, we can talk through it as we draw it.
Yeah, why don't you draw as you guys are talking?
So we had said that the water entered through. I remember you had an arrow.
Yeah, down here at the head of the squid.
Yeah.
If it lets me talk. Is it easier to edit on the max like edit button, or is it easier just to draw it from the Zoom options?
I think I'd probably draw from the Zoom options, but this is up to you.
Yeah, do it on those.
We'll do There we go. We had that. Then we changed this to blue for water. Had it enter through here. And then
The mantle cavity like expands because the stellate ganglion has an action potential that causes it to expand, so water can fill it. And we were kind of confused. We weren't sure if it would have a thin or wide nerve. So that was something that we were unsure of.
You're saying half information. So this is before anything occurs. The mantle cavity expands to pull in water. Is that correct?
Can you repeat that?
So before anything occurs, the mantle cavity expands just to fill with water. So
Okay.
kind of like at equilibrium. Is that what you're saying?
Mm-hmm.
And then like once it reaches that point, there's like some kind of chemical like not reaction, but like chemical synapse.
Well, here, before we step ahead, I'm getting a little confused. I'm just trying to ask a few questions. So one, the head is by the eye, so you have water coming in through the posterior end. Is that correct? Okay, that's cool. That's cool. So why don't we change that? Run this through the posterior.
Let's delete.
And then Natalie, what I was trying to understand is, are you talking about the mantle cavity expands? This is just so that water fills up inside the mantle cavity. So basically at equilibrium, you get water into the mantle cavity.
Okay.
Is that what you're saying? I'm asking.
Okay. Well, that was something that I wasn't thinking about. So if that's the case, if the squid fills up with water before like a predator is near it, then I would say it's a thin nerve. Because we were thinking of it as like a predator is coming and the squid will fill up with water at that point that it notices there's a predator nearby.
Okay. Can you think of what might be the problem with that?
It would probably get eaten because it wouldn't fill up with water probably fast enough, and
so it might get slow, but let's use your logic. Okay, so you're at the stellate ganglion, and can you draw in here? Is this Taylor? Are you doing it?
Yeah, I'm editing it.
Taylor, can you draw lines?
Yes. Let me get the draw. All right.
Okay, so then tell me what's going on here. Why don't we draw lines to illustrate the nerves, and then you can describe what they are like.
Okay. So I'm going to take an idea off this, that the water, you said it enters right here, right?
I know you've got the water entering through the posterior end. Again, this is coming up with hypotheses. These don't have to be perfectly realistic. What they should be is logical. They should be perfectly logical, even if it's not exactly what the squid does.
Okay.
So I'm fine with coming in through the posterior end, you know, yeah.
All right. So do you think it enters in and then like it fills up, like there's a ring, I would say, of water that allows it to expand?
And then through the funnel is where the water comes out.
Yeah. Once it reaches capacity, it exits the squid's body.
Yep.
Right. And then that's the water flow, I would say. Where would we put the nerves, guys?
The stellate ganglion is where the nerves would be to allow for the mantle cavity to expand, because it's like a muscle. So it needs to constrict and
okay.
Does the filling of the mantle cavity need to be coordinated?
Yeah.
In a special way. Okay, well then how would that accomplished? How does it have to be coordinated and how would it be accomplished?
we had said through the chemical neurotransmitters.
Why chemical?
Because that's what I associate with muscle contractions. But maybe the way that chemical transmitters work is,
I mean, that could be a very good idea, but why chemical? So again, what I said is this doesn't have to be perfectly biological in the sense this is what the squid does, but it does have to be logical. So what does the chemical give? And we're just, for your group, we're not going to do anything beyond just filling up the mantle cavity. This is good. So what would the chemical, how would the chemical neurotransmitters help in just filling up the mantle cavity with water?
They kind of indicate when it's met capacity, right?
How?
is it possible that there's like a neurotransmitter that kind of like activates a receptor that can like signal the cell to like bring in water, I guess?
Okay, so maybe there's a receptor which is sensitive to neurotransmitters that sort of bring in water. Now we're getting some good logic. I like this. And what else might you expect with a different receptor? So as a receptor, when stimulated, relaxes the mantle cavity walls, allows water to come in. Yeah, that's clever. I like that. So how do you stop it?
Stop it with another neurotransmitter.
Neurotransmitter?
Yeah.
Neurotransmitter would be the effective way of stopping it. So you use serotonin to open up the cavity and you use histamines to stop it. And I'm just making up two neurotransmitters. It doesn't matter about the function. But you use two separate neurotransmitters.
Yes.
Well,
What gives you more variation? What gives you more control? The transmitters or the receptors?
The receptors.
The receptors do. So based on your description, I think this is actually a pretty good idea. You might have neurotransmitters that are released. And one example is at a certain threshold, the muscle cavity is relaxed, water can come in. And then when the neurotransmitters hit a particular threshold, a new receptor is activated and the influx of water stops. So you're using one transmitter, multiple receptors.
okay.
Yeah, so don't worry about the expel. You guys answered a different question, but I like it. And it's a good way of describing it. Again, what these don't need to be necessarily is exactly what the squid does. What I want us to understand is how neurons can be used to accomplish a task. Now, the great thing about being group one is you guys could kind of change the question and have the liberty to do that. But group two, I want my question answered. And that is, how can the squid coordinate the mantle cavity so it collapses and away from the posterior end to the anterior end and therefore force water out of the funnel? And therefore, Connor, Liz, Millie, Olivia, if one of you guys could share your screen and let's walk through this. And Taylor, thank you very much. You can go ahead and stop sharing.
All right. So our group was slightly different. The question is how to coordinate like the closure of the mantle cavity to expel the water.
Mm-hmm.
I'm pretty much thinking like so it has the connection of from the stellate ganglion to the mantle cavity through wide nerves, so like fast this is like fast action potential. And it will send signals like once it's full, but then like from the wide nerve will send signals from the mantle cavity to expel the water from the posterior to the anterior through the funnel at when needed. Like for instance, when the predator is like approaching, it needs to expel it as quickly as possible. So it would use the fast action potential wide nerves and something like that.
So Connor, where would the widest nerves be? And Liz, Millie, Libby, you guys can answer too. Where would the widest nerves be in this diagram?
We thought it would be in the giant axons,
Okay.
would be the widest.
And where do the giant axons terminate?
In the mantle region.
True, but the mantle region is all along the entire length of the body. So is it in the front of the mantle region, closest to the funnel, the middle of the mantle region, or the posterior mantle region?
The posterior end.
Okay.
Yeah, middle to posterior.
Yeah.
Posterior. And what about these, they're in orange. Are those wide nerves as well?
Yes, that's wide nerves connecting from the stellate ganglion to the mantle cavity. That's at least what we were thinking.
Sure. Where would the widest nerves be based on this logic?
It would be like based off of this logic, the widest nerves would be towards the posterior because you want to expel as much as possible from the posterior to the anterior.
Okay. And based on this logic, where would the thinnest of the wide nerves terminate?
The thinnest would be like closest to like the towards the eye,
Okay.
because you want to press from back in, I believe.
Okay. Does anyone have some questions? So the nice thing about being the second group is you don't have to deal with the questions like what's going on in the stellate ganglion, because you have a logic. Does everyone kind of agree that there's logic here? Why nerves going to the posterior end of the mantle cavity, which means the posterior end of the mantle cavity is stimulated first. It could start a contraction. And then slightly thinner nerves in the middle. So to get that wave from the back. And then thinner nerves towards the front of the mantle cavity. So it waves all the way through the mantle cavity and water gets projected out. Anyone have a problem with that hypothesis? That's pretty good. So now we figured out a mechanism for getting water in. We figured out a mechanism for how to close the mantle cavity. Love it. Group 3 is Casey, Cortland, and Carla. Awesome. If you can make your Word file a little bit smaller so we can see your entire beautiful drawing.
Myself. Okay, now I'm unmuted. Okay, now I'll share screen. Sorry.
Nice.
Yeah, it kind of got cut off anyway, but
okay,
yeah, that was it.
Perfect. Oh, you got some extra stuff going on. So why don't you guys walk us through? And Casey, thank you for showing the screen. Casey, Cortland, Carla, what do you guys got here?
So we have the brain close to the eyes, and I forgot to draw thick neurons from the eyes to the brain, but they'd be thick so when the squid can see a predator, it has a fast response.
Okay, keep going.
And then we have thick from the brain to the stellate ganglion, because that fast response from the eyes needs to be followed by a fast response in the stellate ganglion, because that controls the contraction of the mantle cavity.
Is it one nerve or would you have consecutive nerves?
Think consecutive.
Okay, so what type of junction would be found between those nerves?
Maybe an electrical synapse.
Why?
Because it's faster than a chemical synapse.
Why? Why typically means you have the right answer, just more information, versus really, where I'm not sure that's the right answer, but let's see where you're going and maybe I'm wrong. Natalie, were you going to answer?
Yeah, I had a comment. Maybe because of like it's so close to each other. If it was a different kind, like a chemical, the neurotransmitters wouldn't have to travel. Whether the electrical one, since it's close together, like the receptors, the gates can just like flow through.
Yeah.
I don't know.
There's no translation of the signal. It's just one continuous potential. So it's rapid. So good. I like that. So keep going.
We also said there'd be like thinner nerves to the arms because they just kind of gotta close, which I mean, for me, I was thinking, well, they shouldn't just go like, I know that should go straight, but he may have to turn at the last second. So I figured it was just like, gonna be a slow, like, oh my gosh, you know.
Oh, so maybe the arms have to be a little bit more coordinated where the mantle cavity is just about collapsing. The arms are closing, but there may need to be some more responsiveness in terms of that. Well, that's interesting. So what type of synapses might you expect?
Good question. Anyone else in my group want to piggyback off that?
Would it be electrical because it can be delivered to a specific arm if it's one arm versus another?
Well, all you have to do to get from one arm to another is just have synapses that carry the signal from one area to another. Okay, well, we can think about that. So what's going on in the brain? Why don't we actually focus on that with your group? And why is information sent to the brain, and how is that processed in the brain, and what type of architecture might you expect in the brain?
Well, information's being brought to the brain for processing, and then based on that information, other information is sent out to the rest of the body to generate a response.
Okay. What type of synapses are going to be more valuable for processing? Chemical or electrical?
Electrical.
What type of synapses are going to be more valuable for processing? Chemical or electrical?
Chemical?
Chemical. Why? Oh, I love the way the rest of the class, just like, you guys are like, "Oh, I feel so bad for you." No one's jumping in at all. It's just sort of you guys out on the firing squad.
I'll help you out. Like Carlos said, chemical, because that way it can travel to the rest of the body.
Oh, that's clever, but it doesn't work like hormones. I like that. I like that sort of idea. And that would be a clever idea if that's how it operated. But hormones widely distributed. Neurotransmitters stay very close within that point. They're really going across a very small cleft. So they're released, and then they're sort of picked up on, but still chemical.
Can I add something? Maybe I'm completely wrong, but if it's chemical, is there, and for example, for like the arms bending a specific direction, is there more of variation in like the different kinds of ways you could bend the arm, or like how it's supposed to be oriented? With electrical, maybe it's you don't have such flexibility.
Electrical is on off, so it's like an action potential. Chemical, because you have more than 2 neurotransmitters and you have lots of neuroreceptors, there's the ability to process. Should the squid expel its mantle every time it sees an object? Because an object could be a predator. So just any time it sees an object, it should jump and just flush out its mental cavity and just fly backwards.
No.
No.
An object can be prey. It can be anything.
Ooh, that'd be terrible if you're running away from prey. That'd be a very hungry squid. My family right now is obsessed with the show Alone. And the entire show is just about not finding food. So a squid that just doesn't find food, you could just hear the squid monologue about how hungry it is. That'd be bad. So a neurotransmitter in this case, and a chemical synapse in this case, allows for more processing. So I think it's a good idea. The brain probably does have chemical synapses to differentiate between a dangerous signal versus a benign signal, or a dangerous signal, a benign signal, a desired signal. You know, where a benign signal might be neutral, desired signal might be, as Liz said, food. Great, great. Thank you, guys. Group 4 is Alex, White, Heather, and Megan. Ooh. You guys got some aesthetic in the drawing. I like it. You got like little lines and sort of shows us.
Yeah, like I don't know if we did it exactly the same process as everyone else, but I definitely was thinking there'd be at least this long one here would be a wide nerve, just because it would have to have a fast action. And maybe it's able to like, because it has to push the water through. So maybe this can contort and concave lower, and these little ones can help it do that.
Well, yeah,
and then, you know, next through.
Give me 1 second, because I actually want to focus on your mantle and the nerve you have associated with the mantle, because it's a different hypothesis than what Connor and that group showed, the second group, where what they showed was a series of nerves of different widths going to different parts of the mental cavity, with the thickest nerves going to the back, medium-sized nerves going to the middle, the thinnest nerves going to the front to create that wave. Your group, and I think this is really kind of interesting, Alex, Heather, and Megan, you guys have a single giant axon, and then what looks like additional nerves going off to different areas of the mantle cavity. Is that what I'm looking at?
Yeah.
Cool. So I want you guys to walk through how we can create the same function that group 2 did, Connor, Liz, Millie, and Olivia, but using a different architecture in your design. So how can we create that collapsing of the mantle cavity from the back going forward using your hypothesis? Because it's really clever.
I was thinking that if this was collapsible, I don't know, it just if it had any flexibility to it, that would be something that would help. And maybe Alex and Megan can probably explain it better.
Why have the nerves coming off the side? I want you to focus on that, Alex.
So the nerves off the side, we were thinking like the main highway would be like the spine. So it would be the wide, the wide nerve. But then the little nerves off the end could start with wide at the bottom and then slowly get to thin at the top,
Can I ask for a little clarification what you mean by bottom and top? Do you mean bottom and top the way we're looking at the screen?
Yeah.
Or do you mean bottom and top as the bottom is by the head and the top is the posterior end?
No, so thickest at the posterior and thinnest at the anterior. And so that way, like the main signal is sent down through the spine the fastest, but then once it reaches the individual neurons that branch out from the main one, they react at different speeds, which makes it like a funnel or something.
Do other people think that would work? You have the giant axon that carries the signal uniformly across, but then it connects the dendrites along the axon. And when it connects the dendrites towards the posterior end, those associated nerves are thick. And those motor neurons go directly to the cell wall with a very thick charge. And then as you go closer and closer to the anterior end, the axons coming off that giant axon, the nerves coming off the giant axon are thinner and therefore more slow in terms of communicating that action potential. Does that seem to make sense to people? Is that logic? Because maybe one question, and maybe it's the only question I have in terms of this context, what type of synapse would you have between this giant axon, and I think you've called it a giant axon, you call it the central nervous system. Again, let's call it the giant axon. So what type of synapses would you have between your giant axon and these associated nerves?
I was thinking kind of action. I actually was thinking action.
So which one would give you a quicker action, electrical or chemical?
Electrical.
Perfect. Love it. Anyone got a question with this idea? I think it's good. And I like the fact that it's a different way of sort of solving the exact same problem. So group 5, Dakota, Ella, Shyla.
Does anybody have a picture? I have it, I think. Okay, don't judge, we got bored.
Oh, I love the house of stars. And
okay.
Hola?
Ola.
Oh, of course. Okay.
Yeah,
so
that's you. So we thought that there was like a main giant axon and the smaller ones would go out to the other parts of the mantle cavity because since it needs to all go like uniformly, this one would travel faster. And so it's like start up there and then the smaller, I guess, axons would follow. Does that make sense?
No, it makes sense, but I'm if it's okay, I'm actually more intrigued by a different part of your hypothesis.
Yeah.
Shyler, oh no, this is Dakota, Ellen, Shyler. Oh, all three of you. You guys have this big, huge arrow going from the eye to the stellate ganglion. And you're skipping the brain. What's going on there?
Because it needs to be like a quick anti-predator response.
Okay, so what's going on in the stellate ganglion?
Is that where the action potential is initiated?
I don't know. You haven't told me yet.
That is where the action potential is initiated.
So then what's going on in the eye?
That's where, I guess it started there if it senses some outside information. But the stellate ganglion is where the sensory information is brought to, and then it directs it to the mantle cavity.
So basically the information goes directly from the eye through an action potential into the stellate ganglion, and then it's immediately sent out to the mantle cavity. Is that
Yes,
correct?
that's
So-
what we guessed.
Shiloh and Dakota, why not just skip stellate ganglion? I mean, if that's all that's happening, the information just going from the eye to stellate ganglion and then immediately going out to the mantle cavity, why not skip that step? Because you would be faster. Just have a connection of the giant axon directly from the eye. Hey, Carla.
Well, that makes sense. I don't know. I think I was a little confused during this exercise on what the purpose of the stellate ganglion was. So
Think of it. That's what we're doing. We're hypothesizing.
when we were doing this, I was under the impression that that was acting as the brain
Great. So what should the stellate ganglion be doing then?
processing the information in the eye,
Okay. And what type of information would the eye be picking up?
that there's a predator, there's a visual stimulus of something dangerous.
Is that the only information that the eye is going to pick up?
Well, no. Also that, I mean, in the case of the video, we saw that they were surrounded by other individuals of the same species and everything else around it.
So it's picking up a lot of information. So what might be the point of the stellate ganglion? Shiloh? Ella? Anyone else?
Maybe to, wait, am I muted? Yeah, okay. Maybe to, I guess, determine, can they change their velocity, like depending on where they need to go or what they're responding to?
I don't remember which group did that, but they can change their direction based on how they use their tentacles. And it was one of the groups that had some neurons going from the brain in their hypothesis to the tentacles. And I think Casey, you were talking about this and how they might use that to help orient. And that was clever. That was clever. But you guys don't have that in your hypothesis.
Well, we have this neuron. I don't know why it wasn't curved. That's just what we could do. So potentially, maybe instead of it going directly to the stellate ganglion, this is like, I think we did mix up the function of the brain with this, and we like determined that to be where information is processed, but maybe that would connect the function of the stellate, the mantle cavity to the tentacles.
Can everyone
give coordinated?
Sure. Can anyone give me a definition of a ganglion?
A bunch of nerves.
Bunch of nerves. Can anyone give me a definition of a brain?
A bunch of nerves.
Like a bunch of nerves. That's really what we got. With the brain, we got more nerves. With the ganglion, we got less nerves. We just got a bunch of nerves. And why would organisms have bunches of nerves together?
Maybe to organize an action potential.
Organize actions. And therefore, to organize action potentials. It doesn't always have to be the brain. Even us, we have ganglions going down our spine that help for different types of processes that you don't necessarily have to organize necessarily in the brain. So no, the idea of going straight to the stellate ganglion, that's a reasonable hypothesis. The idea is then to organize instead of what I was thinking you guys might talk about, which is sort of organizing information it gets from its environment and whether to respond or not. You guys use a different tactic, which is fine. And that is to organize the type of neuronal responses. where the nerves up at the tentacles are doing one thing, and the nerves going back to the mantle cavity are doing another thing, and to sort of organize that, I'll call it logistics. Is that fair? That works. I like that. You guys go ahead and stop sharing. So in this, it wasn't necessarily about, and what I want you to take from this, it's not necessarily about knowing exactly what the architecture is. That's it. That's easy enough. What I want you guys to get some practice in is understanding how the architecture can work together to accomplish specific goals. And the behavior in this context is the goal. And therefore, it's got to be accomplished in a way, and there are different ways of doing it, even with such a simple system like a neuron, which is so simple. Any questions with that? Okay, I want you guys to take a 4-minute break. I know we're in the last 20 minutes of class, but I've got a little bit of like staring back at Socrates thing going on, and I feel like there might be a killing in a few minutes. So give yourself 4 minutes. I'll call you back at 1. There you are. So we have a poet coming next week who's going to give a reading on Thursday, who has worked in a place called, what is it? Abkhazia? And I thought I was pretty familiar. I thought I had pretty damn good geography and knew my stuff. Do you know that there are 4 frozen state republics that came into existence after the breakup of the Soviet Union? That, I don't know, they're basically in this perpetual warfare. So like this country is a little chunk of Georgia that Georgia doesn't recognize, but Russia says, no, it's there. There's this little river valley along Moldavia. It has a perfect name. What is it? It's Transitaria, which is awesome. It just means like you walk over it. I think you do it in 1 step too, maybe 2. Who knew? Did you guys know about this? 4 of them. 3 of them are in the Caucasus, and 1 of them is right across the Ukrainian border. Crazy, crazy. Okay, so let's get to the last wonderful 15 minutes. We got that. We got this. Who knows? Maybe I recorded that 3 minutes of me just looking at Wikipedia and reading about Transitaria because that was just cool. So, bats.
If you'd asked me 10 years ago when I started my lab that I'll have a 1 kilometer tunnel and recording from a whole colony of bats, I'll tell you you're nuts. I'm studying the hippocampal formation, a set of brain areas that have to do with learning and memory. And I'm studying those brain areas in somewhat unusual animal models in bats. Bats, I think they're very cute. The bats that we work with, the Egyptian fruit bats, have these interesting behaviors that we try to utilize and that give advantages over the standard animal models, which are mice and rats. So for example, if we want to ask questions about how 3D space, 3-dimensional space is represented in the brain, bats are flying mammals. So obviously they're a good choice for that. We can also, another advantage is that bats have 2 distal sensory systems, vision and echolocation. We can trade off these 2 senses and ask how the same space is represented under these two systems, which are difficult to do in rats or mice. We also ask questions about how very large spaces are represented in the brain. So we've built this long tunnel, 200-meter long, and we are building this year an even longer tunnel, almost 1 kilometer long. And we have bats flying there 100 or 120 times back and forth while we record neurons in their brain, and we see very different representation in this enormous space. we implant these small electrodes in the bats that allow us to record the activity of single neurons in those brain areas. And we developed these small wireless electrophysiology devices called neurologgers that are really small and sit on top of the animal, which allow us to record wirelessly in a bat as it's flying around. And then we can have them fly in a large flight room or in various large-scale spaces that we've built and study the representation of space. We can also have bats interacting and ask more social questions in the bats. What I'm really interested in is the neural basis of natural behaviors. You know, neuroscience have traditionally focused on either, of course, anesthetized animals that don't behave at all, or if they're doing behaviors, they're doing highly controlled behaviors, lever pressing or nose poking in some very 30-centimeter size box. The big plus side of it is that it's highly controlled, so you can get very repeated and very controlled experiment. The minus side of it is it's very far removed from natural behavior, from what the brain evolved to actually do outdoors. And so I really believe, in addition to having those highly controlled experiments, we should ideally record the same neurons, or at least from the same brain areas, from an animal behaving in a more natural situation. Now that the technology allows it, that we have technology for wireless electrophysiology, for recording for freely moving animals, from wireless optogenetics, camera arrays, with microphone, with movement sensors, all sorts of devices were not available until a few years ago. We can really study the neural base of behavior for the first time, for real.
All right. A few reasons I wanted you guys to see the video. One, many times the ability to ask the question depends on finding the right animal. And a lot of times we just sort of just quickly go back to something that is easiest, like a lab rat, which is great. But in order to address certain questions about actually how you perceive larger landscapes, this does a much more effective job. Two, we're going to keep with those themes of bats, and we're going to look at a little neurology within bats and their prey. And then three, if you ever had the impression that scientists are half in it for the toys, the answer is yes. That was cool. A kilometer-long flight chamber in a brown field in the middle of Eastern Europe, so you can fly around Egyptian fruit bats. Yeah, that would be an awesome thing to have here at FGCU. Questions? Okay, so I want to end, I think, maybe with 2 or 3 more slides. We've talked about bats in the past, and we've talked about their prey in the past, and we recognize the fact that we have sort of a fairly straightforward mechanism that these bats are using in terms of echolocation, and that they could sort of call out a very high energy, high frequency sound, and that can produce precise echoes which allow for the bat to find its prey. We've talked about some fairly cool and elaborate ways in which prey can avoid predation, like the sonar jamming tiger moths, where they will produce an ultrasonic call that disrupts the echoes, and therefore the bat can't find it. But the most common way for non-tethered prey, and this poor moth right here is just attached with a line, is when they hear that echolocation, they just drop. Now, dropping is key, because by dropping, the bat has to reorient, but the moth has to do a variety of things, and that has to be able to hear signals, know the direction, know when it's coming, and if it just drops all the time, it's going to have to use a tremendous amount of energy to get back up in its flight. It's going to waste effort, waste time, waste resources. So, like most things, it has to pick up on signals correctly. Well, in insects, they have ears, and these ears are called tympanums. So it's an ear. And basically what it is, it's a flat surface, much like an ear that we have, where there's an air sac, and this flat surface will stimulate, in the context of the antepredator display of noctuid moths, one of two possible cells, A1 and A2 receptor cells, where these are sensory cells right along the surface of the bantam, and when this sac of air is vibrated, that is hearing a sound, and among other things, these two cells might fire off. How these cells fire off, when these cells fire off, provides these moths with a tremendous amount of information. So when the stimulus of a sound, an ultrasonic sound, is low intensity, then the A1 cell will fire off with semi-frequency relative to when there is no low intensity sound in the background, where the A1 cell will just fire off at a regular interval. At the same time, where you have a low intensity sound, the A2 receptor really just fires off at a very regular, slow interval. When the intensity of the sound, this ultrasonic sound, becomes more intense, moderate intensity, A1 starts to fire off at a higher frequency. When the sound dissipates, it's no longer being produced, then A1 fires off that normal interval frequency. At this moderate intensity, A2 is still firing off at this normal moderate frequency. There's no change as a result of the high intensity sound. When the sound, though, becomes very intense, i.e. it's loud, there's a lot of energy in it, those air sacs are shaking intensely. At this point in time, the A1 sound, the A1 receptor is still firing off at a very fast frequency, where each one of these lines represents an action potential. When the sound is gone, it's back to that regular interval of an action potential, delay, action potential, delay. When the sound is highly intense, against those air sacs that are really shaking, then at this point in time, the A2 receptor also starts to fire off very high frequency of action potentials. And then when the sound dissipates, it goes back to that regular interval. So in low intensity sounds, a few action potential in A1, none in A2. Medium intensity sounds, some action potentials in A1, nothing in A2. High intensity sounds, a lot of action potentials in A1, a lot of action potentials in A2. In addition, the A1 receptor shows a pathway by which, when it hears an ultrasonic pulse, it will produce a lot of action potentials. And then when there is a trough and it hears another high intensity pulse, it produces a lot of action potentials. A trough, nothing. A pulse, a lot of action potentials. Trough, nothing. A pulse, a lot of action potentials. By comparison, when there's a steady, high ultrasonic sound, you'll see a lot of action potentials initially within the receptor cell, but then that starts to dissipate as the action potentials go down to that regular interval and the animals habituate. Make sense? Okay, what I want you guys to do, and we're going to wait to the beginning of class on Tuesday, as we're going to address this question. Now, on Tuesday, what else are we doing?
A peer review.
We're doing a peer review. And at this point in time, by Tuesday, how far along should you be in your biographies?
Mostly done.
Not mostly done. I mean, if you can, oh God, that'd be awesome, Taylor. That'd be great, because then you have that many more chances to sort of edit, refine, improve. That would be fantastic. But I would say a really good place for next Tuesday is to have revised the personal history and the topic introduction based on what you guys have received, and then to have a good functional working draft for the 3 hallmark papers, the early career, mid-career, and late career. Remember, you're not describing every result, you're describing some results, the most pertinent results. And you may want to have sort of some outline or some ideas about the overall work history of this researcher that's sort of synthesizing the stages of their career. and maybe some ideas about how this person is impacted. But if you could get to the point where you have a good functional working draft of the 3 papers and you are set with that personal history and you are set with that topic introduction, you're in great shape. Make sure that you submit a copy onto Canvas because I will be taking a look at it. Make sure that each person in your group has a copy for themselves. And then just like last time, you get full credit for submitting a draft and you get full credit for being part of the peer review. Any questions? Have a great weekend. Do something fun.
Only one person from the group needs to